Hello, everybody. My name is Abhinav Yamujala, and this is my presentation on the topic of Tay-Sachs disease, its relation to global health, and my approach to solve this problem, which is the title of this slide, a synthetic biology approach for an AI-optimized protein cloning to mitigate the progression of Tay-Sachs disease. So a short introduction about me is I'm a rising senior, and I'm currently attending Lambert High School in Atlanta, Georgia. Some of my notable extracurriculars include being part of the iGEM seminar team, being part of my robotics club, being a researcher at NASA Gene Lab, and also being the president of my school's local science school chapter. In the future, I hope to pursue cardiothoracic surgery or do something along the lines of computer science and biology. So let's start off with a little background information on Tay-Sachs disease. So Tay-Sachs disease is characterized by a mutation in the HEX-A gene, which leads to an underproduction of the protein beta-hexospermidase A. So the production of beta-hexospermidase A is important because beta-hexospermidase A is responsible for preventing the buildup of GM2 gangliosides in the central nervous system. Without it, it leads to a buildup of GM2 gangliosides, often leading to the body to being unable to function by itself. So almost 1 in 100,000 babies are affected worldwide. And although this may seem like a small proportion, it's actually a large proportion of our population. The average lifespan of a child with Tay-Sachs disease is only 2 to 4 years old. Despite the large amount of people that are affected by Tay-Sachs disease, there's little to no research being done on Tay-Sachs disease. As you can see in this diagram here, there's only 0.7% of research, of neuroscience research dedicated to Tay-Sachs disease, and similar rare neurodegenerative disorders like Tay-Sachs disease, like MELAS, which has only 1% of research contributed towards it, are prove how these rare neurodegenerative disorders are being neglected in today's society simply due to their rarity and due to their complexity. You can also see here how several underdeveloped areas in the world do not have access to genetic testing to basically diagnose themselves with Tay-Sachs disease, or to test whether they have Tay-Sachs disease, or to mitigate the symptoms of Tay-Sachs disease, because they have inadequate access to genetic testing and gene therapy. So some examples here, you can see Latin America only has 3% of basically genetic testing interest, and Africa has almost little to no genetic testing available. So this leads to the next topic. Tay-Sachs is basically a global health problem. Because there's limited access for genetic testing for Tay-Sachs and several other rare neurodegenerative disorders in underdeveloped countries, this leads to no treatment, and therefore several scientists are not investing their time to actually study Tay-Sachs disease and other neurodegenerative disorders and find out whether there's a cure to these rare disorders and whether there's some way of mitigating the symptoms. Because of the lack of gene testing, there is then a misdiagnosis, and this often leads to a never-ending cycle. So let's start off with the causes and symptoms of Tay-Sachs disease. So the cause of Tay-Sachs disease is basically GM2 gangliosides buildup, because as we said before, the beta-hexospermidase A protein is underproduced, and therefore the GM2 gangliosides cannot be broken down. And because Tay-Sachs disease can be caused by over 210 unique mutations, again, several researchers are not dedicating time or investing time in order to solve this problem. And Tay-Sachs disease is often inherited by the parents to their offspring. So symptoms that are seen in Tay-Sachs disease are often seen in children aged 2 to 4 years old, and this can be anything from vision and hearing loss to severe seizures. So an example of this is seen in the X-ray diagram below, where it shows an individual with Tay-Sachs disease who has an enlarged head, showing how Tay-Sachs disease can also cause facial deformities. So my approach to solve this problem was basically using my expertise of artificial intelligence and synthetic biology. So I used synthetic biology in order to create a plasmid construct, which I basically used PUC19, which is just the base of the plasmid, and added simple parts such as the operator and promoter regions that are specific to HEX-A in order to overproduce HEX-A and account for the underproduction of the beta-hexospermidase A protein. And then I also used a SDS page gel electrophoresis and a Western blot to ensure that my plasmid worked and to ensure that I wasn't overproducing the HEX-A gene too much. So for the MPRA array, I also used artificial intelligence in this project through MPRA arrays, which are a type of machine learning algorithm. This basically further evaluated the plasmid effectiveness. And how I used this was I measured the amount of RNA produced after introducing the cell to the plasmid. And then I also basically measured the DNA produced in order to evaluate whether the plasmid actually worked and to make sure that there were no off-target effects happening. So the second kind of problem that I encountered in this subject was finding a way to effectively deliver this plasmid to the brain directly. So I decided between two options, nasal spray or injection, and I decided to do a nasal spray. This is because a nasal spray is not only extremely harmful, but it can pass through the blood-brain barrier with no off-target effects due to its high specificity. And this is by traveling basically through the trigeminal and the olfactory nerves. On the other hand, if I want to do an injection, the injection can only be administered to the spine, which is not only extremely expensive, which basically restricts access to underprivileged areas, but its low specificity can also lead to several off-target effects because it may be misplaced on a nerve that is central to the body. So the future plans for this project is that I want to hopefully test on mammalian cell lines and create a off-the-shelf therapy, which is basically I want to be able to sell it to pharmaceutical stores and make it equally accessed across the world. I also want to kind of lower the number of base pairs for the plasmid. There are almost around 2,600 base pairs. So I definitely want to lower the amount of base pairs in order to make it more cost efficient and more easily accessible in underdeveloped regions. So through this project, my main goal was to basically not only create a cost efficient treatment for individuals with Tay-Sachs disease, but I also wanted to bring attention to several neurodegenerative diseases like Tay-Sachs disease and other rare neurodegenerative diseases that are often not studied in the research academia. So this is a research manuscript. I was able to present this at the Ragan Institute of MIT and Georgia Tech, and it is pending a publication in the Biotrics Journal. These are my references. And I would also like to thank Dr. Alyssa Morrow for helping me with the AI model. And I also want to thank my school advisors, Mr. Scherer and Mr. Schuyler, for helping me basically make this project feasible. I would also like to thank my teammates in the iGEM seminar team for helping me with the development of the idea and the execution of the idea. Thank you for listening. This is my contact information. Feel free to send me any questions you have about my project, and I would be happily able to answer them. Thank you so much.